Hello class, this is our asynchronous video lecture starting from where we left off on class on Tuesday. So the last thing we did was we watched this delightful video of the superb lyrebird, a bird that is found in Papua New Guinea, northern Australia, actually throughout Australia I think. And so we were talking about the avian song learning system, and in many taxa of birds, especially ones that we're all fairly familiar with, whether you know it or not, songbirds. So your classic backyard birds like cardinals and sparrows all have song learning. And this behavior of singing in birds, which is making vocalizations that are relatively complex and long, is largely a sex-specific behavior among temperate migratory songbirds, like your sparrows. And so the differences in behavior are underlined by differences in neural architecture and hormones. So let's talk about those mechanisms. So within the avian brain, there are specific regions that mediate the production of song. And these brain regions differ between the sexes. Let me move me up here. So let's take a look here at some of these. So RA, this stands for the robust nucleus of the archistriatum. And as you can see here, this arrow is pointing to a slightly darker staining of the neurons of this part of the brain. This is the RA in a male typical brain. Contrast that to a staining cross-section of a female brain. It's much smaller. And if we take a look at the map of all these important areas in the brain, and typically collections of neurons in the brain that serve that are involved in a particular function, particular processing function in the brain or network, they're usually referred to as a nucleus or nuclei in the brain, which is different than how we refer to it in cells. Right? So when I say nucleus, talking about the brain here, I'm talking about a collection of multiple cells, multiple neurons that all kind of do the same thing, rather than, the middle of a cell that has its own membrane that contains DNA. It's a different type of nucleus. So, for example, let's take a look at the HVC, which is the high vocal center of the avian brain. Very important in producing song. And so this upper one is a male zebrafinch, a graphical representation, and this is a female zebrafinch. Female zebrafinches don't sing, typically. The numbers here and the relative size indicate the percentage of neurons that have androgen receptors, specifically receptors for testosterone. So in males, it's about a third. A third of these neurons have receptors that bind testosterone, whereas in females, it's closer to about an eighth of the cells, 12%. As you can see, there's a number of different regions here that are important. I'm not going to require you to memorize these regions in the song learning network of the avian brain. But if you really want to, you can take my ornithology class in the spring, and I'll make you memorize it. Oh my goodness. What a disaster. Back on track. Okay. And so the differences in the neural structure that we see on average between male and female songbirds is due to organizational effects during early development. Let me make sure I didn't miss a slide here. Okay. But there's also androgens also serve as have activational effects. All right, so the singing itself, if you have this neural architecture in place, if you have these relatively large regions that have a robust percentage of androgen receptors, you're primed, you're ready to produce song, but you still need androgens to produce song. And that's why we see such a high density of testosterone receptors. But we shouldn't forget that estrogen also plays a role in this song production as well. It's not just androgens. On the right here, I've done showing you an image where they've stained the actual receptors themselves in the neurons in the high vocal center of the brain. And so here's another graphical representation to show you the distribution of the androgen receptors and the estrogen receptors. And so the role of estrogen in producing song in the HVC, high vocal center of the avian brain, is a relatively recent discovery, and it's just another testament to how there is really no such thing as a male hormone or a female hormone. We all have both, and that's typically across taxa, whether or not you're a mammal or a bird. Now we know that androgens are important in the organizational effects of the avian song learning because we've demonstrated that we can experimentally produce singing behavior in females. And so this has been done in European robins, canaries, a handful of other species, where female songbirds that are treated with testosterone during development, during those critical windows that I've talked about, they exhibit later male-like song nuclei, or they exhibit male-like song nuclei in the brain. And if you give them an injection of testosterone, it will activate that architecture and they will sing, very similar to males. Now, interestingly, before I get to the interesting part, my battery's going to die. So, take a break. Talk amongst yourselves. Okay, back on track. So quite interestingly, the organizational effects of testosterone are actually due to estradiol. So I've changed our title here a little bit. Organizational effects of estrogens. I've crossed out androgens. Because as it turns out, it's actually estradiol that's important. And so you may remember before, I've mentioned it a few times, that estrogens, specifically estradiol, which is the most common estrogen in both mammals and birds, estradiol is converted from testosterone via an enzyme called aromatase. Right, so this is an important enzyme to put in your head. Aromatase is how we get from testosterone to estradiol. And as you can see here, the similarity in function is pretty remarkable. Right, we've got this carbon backbone that you see that's very similar to cholesterol, because all of these steroid hormones start with cholesterol, and they sort of work their way down, converting from one to the other via enzymatic actions. So what's the story here? I've been telling you this whole time that it's androgens and testosterones. Why am I switching now to estradiol? Well, let's take a look and see. So imagine the avian embryo in the egg. It's sort of developed along. It's got its testes, which is pumping out androgens. Its clutch mate, its sister, doesn't have this. They don't have testosterone floating around in their system to nearly this concentration because they don't have testes. So these male embryos have testosterone surging through their circulatory system, and it's going everywhere, including to the brain. It's getting into neurons, and once it gets into the interior of the cell, right? So there's some little carrier protein here that the androgen receptor that it attaches to, and it actually transduces through into the cell. And then aromatase is in the cell in quite high concentrations in neurons in these vocal centers of the brain. And it converts testosterone to estrogen. Estrogen, specifically estradiol, and it attaches to an estrogen receptor, which then allows it to go into the nucleus and attaches to DNA and serves as a regulatory element in gene expression. And so what do we call that? What is this complex of estradiol and this protein regulating gene expression? Anybody? Transcription factor. And so this altered gene expression, what it does is we follow these arrows here. This is the transcription factor. This little arrow indicates that it's mediating transcription of different genes. It changes, right, alter gene expression. And what it does is it downregulates the cellular process called apoptosis. Apoptosis is cellular death. So what it's doing is it's downregulating it. It's stopping it. Estrogen, by and large, has a protective effect. It protects neurons from self-destruction. So as we see in reality, though we think of testosterone, masculinizing the brain, really, technically, it's estrogens that are doing it, because testosterone gets converted to estradiol via aromatase. And like I said, this transcription factor acts to regulate cell death, apoptosis, or survival. So one area of the brain that's sort of a big one in terms of being different between, on average, between male and female individuals is the sexual dimorphic nucleus of the preoptic area, SDN-POA. And so neurons of this area, like in the songbird song learning network, the SDN-POA is rescued from cell death by the actions of estradiol, specifically the ones that have been converted from testosterone inside the nucleus, or inside the neurons. And so the SDN-POA is a sex-specific, a part of the brain that differs between the sexes in mammals. And so a lot of research has been done on this part of the brain in, as you might imagine, mice and rats. So in both songbirds and that in songbird, in the song learning networks, and the sexual dimorphic nucleus in mammals, it's the males that have the larger part, that express larger areas of those. But that's not always the case. It's not males that always have the larger parts of the brain. And here are some examples. The anterior ventral preventicular nucleus, AVPB. I'm not going to. You don't need to memorize this. You don't need to memorize the AVPV, largely because you can have an open note, open book, open resource exam. So I would say don't spend a whole lot of time trying to memorize these parts. And so this area of the brain, the AVPV, is involved in ovulation, regulation of reproduction, and it may be no surprise that it's larger in females, in both rats and males. And we can see here staining of nuclei in the nuclei of neurons in the AVPV, and they're many more and much more dense in female sections of the female brain compared to sections in the male brain. And so in this example, in the AVPV, it's testosterone directly induces apoptosis, which causes the reduction in its reduction in size in male brains. So the testosterone is actually causing cell death. And who has all the testosterone during early development? Well, it's those embryos that have testes. It's the males. So androgens beyond mediating cell death and survival, they can also act as neurotrophic factors. So these are molecules that mediate, facilitate growth, survival, differentiation of both developing and mature neurons. So let's take a look at some examples. So during development, estradiol can increase physically the cell size. So here's an example where they took parts of the hypothalamic explant. So they're basically taking out some neurons from the brain of either a rat or a mouse here, putting it in a growth medium. This one had no estradiol, and this one had estradiol. You can see that the neurons are much more robust. The hypothalamus is an area of the brain, part of what we call the limbic system. In the limbic system, or specifically the hypothalamus, is involved in a number of different things, including connecting higher parts of the brain or the neural system in general with the endocrine system. It's involved in kind of physiological processes and behaviors that aren't associated with our big, large cortex, those types of things that we kind of that we think about that makes humans so smart. So it's involved in things like the autonomic nervous system, reflexes, metabolism. It helps regulate breathing, heart rate, our circadian rhythms, hunger, right? Things like that. That's what the hypothalamus does. If you want to learn more, you can take neuroscience in the spring. Let's see, estradiol can also alter the hippocampus. And the hippocampus is another specialized part of the brain. It's very, very important in learning and memory. All right, so you might have heard stories about, really cool stories about New York City drivers. There's been some pretty cool neuroscience studies following New York City cab drivers from when they started until a later date, doing MRI imaging and tracking the size of their hippocampus. And their hippocampus literally gets bigger after being a New York City cab driver for a certain amount of time, because it's important in spatial memory. Right, no surprise. So, right, estradiol is important in the in sort of this plastic flexibility of the hippocampus. One of the things it does is that it facilitates synaptic connections. So that's when, we'll take a look, this is our little neuron here. This is a single neuron, a cell in your brain. And what we have here are called dendrites and axons, and these are basically, you can think about branches, and they touch other neurons, and that's how they send information back and forth to each other. So you might imagine if the neuronal network in your brain of all these different axons and dendrites touching each other, sending more information, if there are literally more connections, you have a higher processing ability, so to speak. So estradiol can facilitate the growth of synaptic connections. And there's been quite a bit of experimental evidence for this, and so let's take a look at this in the rat brain, specifically associated with the estrous cycle, right, the reproductive cycle in rats. And so let's follow this purple line through the estrous cycle. And this line indicates the endogenous estrogen, specifically estradiol concentrations. And so as we go along in proestrus, we get the highest peak, and this is associated with the highest receptive behavior of female rats. This is just prior to ovulation. And right here at the transition between proestrus and estrus is ovulation, right here at this boundary. And so an egg is ovulated, estrogen starts to go down and reaches a low point, and then there's these later stages, medestrus and diestrus, which is sort of like the down phase in the cycle. And so, if you've had pets before, if you've had an unspayed cat or dog, it's these areas, especially proestrus, which is what we would call heat. And so we talked about that a little bit in cats earlier in the semester. That sort of behavior that's associated with sexual receptibility. No, that's not the right word. Reception. Largely driven by this increase in estradiol. So I've got the different phases here coded by letter. And this is a close-up example of an axon. And you can see right here all the little spines coming off. Right? So each one of these would be connecting to another neuron. And so this is A. So this is what it looks like close up during diestrus. When we leave diestrus, as we go up this hill of estrogen secretion, reach its peak near the middle of proestrus, we can see that the synaptic connections increase dramatically. And estradiol is the mechanism by which this happens. Again, like apoptosis, it forms a transcription, it becomes a transcription factor in the nucleus, and it mediates and regulates gene expression, specifically genes that end up producing these higher density of synaptic processes along the axons and the dendrites. And as estrogen falls during estrus, we can take another snapshot of the axon, and we can see that the density has decreased quite a bit in response to the lower concentration of estradiol. Right, so this is a process that's taking place in rats over the course of days. Okay, I can't remember exactly what the estrous cycle is in a rat, but it's close to a week, 6 to 10 days. So this is quite a impressive amount of neuroplasticity. Very, very old way of thinking about the brain is that, once you become an adult, your brain is kind of fixed, and then you just slowly lose neurons over time. And that's not the case at all. Everyone watching this video, you, me, in the world, our brains are constantly growing and changing. And steroid hormones play a very important part in that process. So here's a very important question that I want to ask. We've talked about the importance of steroid hormones, especially the organizational effects, which typically happen during critical windows during early development, like in the songbird for singing. If you block testosterone in a male during early development, it will never develop those robust nuclei in the song learning network. And it doesn't matter how much testosterone you pump into that male. If he doesn't have the neural architecture, he's never going to sing. So this raises an important point. If testosterone is so important, and technically it's so important because it gets converted to estradiol and regulates apoptosis, why doesn't estradiol from the maternal ovaries or in the maternal yolk of an avian egg, right, masculinize female offspring? So I want you to think about this question. We're going to discuss it in class on Friday. And so this is a big one. This is quite a paradox, right? If estradiol is regulating apoptosis and it's leading to specific areas of the brain, if it's protecting specific areas in the brain from cellular death, why does this not happen in females? Right? Especially in mammals, where in utero, right, they're sharing a blood supply. They're sharing the maternal blood supply through the placenta. Right? The estradiol that the mother produces from her ovaries is circulating in the embryos that she is gestating. So in a rat, she's got a litter of pups of different sexes. Their circulatory is bathed in her estradiol. So why do we get these sex-specific differences in neural architecture? So think about that. See if you can come up with a hypothesis. There is an answer for it, and we'll see if we can't get close to it. Okay. Thanks, and I'll see you on Friday.